All right, so I'm happy to be here. My name is Kristien Verreydt, and I work as a client partner at Radix. I'm passionate about helping knowledge workers do better work, and I believe AI has a great opportunity to help here. I'm presenting today our collaboration that we did together with pharma company GSK, where we help lab researchers reduce the time spent on tedious manual work so that they can get results faster. And as Joel already mentioned, this is a really hot topic at the moment. So what I will talk about today, I will quickly introduce what we do at Radix, how we view AI as a co-pilot, and specifically how we view the role of AI in the pharmaceutical sector. I'll show you our 3 pillars on which we base every AI project and how it allowed us to achieve success together with GSK vaccines. And lastly, I'll introduce some other low-risk, high-impact opportunities for AI, which we identify together with our pharma clients. So for those who are not familiar with GSK, GSK is a global healthcare company with the purpose of helping people do more, live longer, feel better. They have 3 global businesses that research and develop vaccines, medicines, and consumer healthcare products. As for us, Radix, we're an AI solution provider based in Brussels, Belgium, and we help organizations build and scale AI solutions. We do this in 3 ways. We help organizations identify AI opportunities on a more strategic level. We build custom AI solutions for clients, and we help teams build their AI expertise through coaching and training. We work for a variety of sectors, including life sciences, but also, for example, media or HR. So how do we see artificial intelligence? We start from the definition of AI as the automation of tasks that require some level of intelligence. So this really means putting AI in the role of a co-pilot to work together with the experts to get better outcomes. So this could be reducing the time spent on a certain calculation or task, improving customer experience by responding to queries faster, many other topics. Specifically, what could AI contribute to the pharmaceutical industry? When we look at the drug development process, we see a number of different problems. The first one is that it takes years or even decades to develop a drug or vaccine. And the second one is that the cost of the development of a drug or vaccine is extremely high. At the same time, there are many manual repetitive tasks that need to be done that are peripheral to the actual drug development process, but that are more administrative. And there is where we as an AI company with a focus on generating impact are so interested in improving these areas. So we believe that AI can reduce the turnaround time and costs in the drug development by taking on some of these manual, mostly administrative tasks so that researchers can focus on what really matters. Let's move on to our use case. So GSK approached us about this step in the development of vaccines, where the lab needs to process hundreds of images that look something like this. So images of Petri dishes. And what you see on this image are bacterial colonies. These are used for one of the common time-consuming and error-prone tasks in the labs, which is the counting of these bacterial colonies. Now, where do those images come from? The lab scientists, in order to correctly quantify and characterize the production of a vaccine, need to take samples of the proteins that they're extracting to eventually create a vaccine. And they need to take those samples at different stages to see how the bacteria are growing at any given time. Now, if you're familiar with the pharmaceutical industry, you might know that there have been cameras around for a long time that do exactly this type of work. And in a lot of cases, they work fine, but in a lot of cases, they also still fail. And this is why an additional development was needed. Every dot that you see on this image is a colony forming unit, or CFU. The lab technicians have to annotate these images. So annotating means identify these colony forming units as either virulent or avirulent. On the screen, you see now the annotation of these CFUs with the categories indicated as different colors. GSK asked us if this process could be partially automated by AI, and that's what we did in the project using computer vision. The way that we at Radix ensure project success is according to 4 pillars. We use these pillars in every single project that we do. So these help us align on the business case, enable the people, enable the end users and sponsors that we need to guarantee project success. And we build AI applications that are usable from the start. In the first step, we had a discussion with GSK on how we could measure the impact of this project. Because if we know how to measure the impact, we also know what we need to do to maximize the impact. We spoke with the lab, and together we identified 2 goals for the project. So the first is that we want to reduce the amount of human time spent on the counting and annotation of these images. And the second goal was to improve the accuracy of the analysis. Once we understand the goal, we can move on to understanding the end user. By taking the time to get to know the end user, we want to make sure that we build applications that are useful, that end users trust, and that they also want to use. And to find that out, we conducted interviews with lab technicians who would later on go off to use the application. And we came to a few important conclusions. The first is we found that most lab technicians don't find this task very exciting. It's very time consuming, and it goes hand in hand with a lot of peripheral tasks that don't really require expert knowledge, like looking for the files, uploading them, reporting back on the results. And finally, we also found that lab technicians generally don't even agree on what the correct CFU counting of a lab sample is. Next up, now that we know where the most impact can be made and what our end users are looking for in the solution, we can move on to building a first working version. At Radix, we call this first working version a skateboard, a solution that doesn't have yet the most complex technical setup, and doesn't have an advanced machine learning model yet, but something that can already get you from A to B. And that way, end users can interact with it, give feedback on where improvements are the most needed. After the first sprint, this was the skateboard solutions that the end users got to use. So what you see happening right now is someone is uploading a snapshot of a petri dish, and in the background, the AI model is predicting the category for each colony. Users can now provide feedback whenever necessary, and this is very valuable because we can show to the end users what kind of solution they could get, and at the same time help them steer the development to make the solution answer their frustrations better. For those interested, here are some of the technologies that we use to build this solution. Now that we have our first working version, we can take the next few sprints to iterate over the solution a number of times until we get to a version that the end user is satisfied with. And that solution looked something like this. So the images are now processed in the background. You will see now as well that there is a zoomed in version that helps the researchers validate the results better. The solution is now also aware of different dilution factors. And finally, the user can now also easily export the results of the analysis. Now, let's have a look at the results. As I mentioned, we wanted to reduce the amount of time spent and at the same time improve the accuracy. The first thing we did was measure the amount of time it took to count the CFUs with and without the solution. And we see that on average, using our solution is 6 times faster than doing the counting and annotation by hand. So just for reference, this is a process that generally took about 35 minutes for one petri dish, and we could reduce it to about 5 minutes. So even in a process that it's still not fully automated, there is still always a dropout of samples that need to be analyzed by an expert. We can already see that such an assistant can offer enormous time savings. We've also measured the accuracy of the automated counting, and there we were able to conclude that the application achieves superhuman accuracy. Now, what does that mean exactly? So this means that the number of times when the expert agrees with the analysis of the machine is higher than the number of times the expert agree with their colleague. Of course, CFU counting is just one of the manually heavy tasks in the drug development process. And together with our life sciences clients, we have brainstormed on where else AI can generate high impact in this process. For this brainstorming, we use a framework called Fast Discovery, which helps us identify the high impact use cases and helps us and our clients prioritize these use cases. When we look at the drug development process, I've shown you this image before, it looks like a funnel. When we start at the top of the funnel, we're discovering potential targets. Could be 10,000, could be lots of compounds. And we want to validate or reject these as soon as possible so we don't lose precious time and resources in testing these further along in the process. So CFU counting, as I discussed, is just the start of this process. And before we can move down to the next stages, for example, clinical trials, manufacturing of the drugs, there are a lot of reports that need to be submitted and approved. And again, this is not one of the most loved jobs done by lab technicians and researchers. So this is a great moment to introduce techniques like natural language processing. So for example, we could gather all the information that we need to submit to get approvals. And this is for this, we can use a task called information retrieval. We can classify the documentation to more easily compile reports. And that uses a task called document classification. We could also, for example, look at recommender systems to propose the most relevant documents for you to look at. We could use natural language processing to compile reports faster. For example, by using text summarization, we can automatically compile pieces of text to use in our reports. And in a regulatory context, we could use AI to make sure that all the information we need is there. And for that, we could use a technique called information retrieval. And of course, it doesn't stop there. This is just a highlight of everything that is possible in terms of saving time and effort in the tasks surrounding drug development. AI can do so many more things to help people doing their best work. And that brings me to the end of my showcase today. I hope you found this interesting, and I'd be happy to answer any questions that you still have. Now, before I go though, I'd like to mention a few things. If you check out our Radix booth, you will find a detailed case study on the CFU counting case that you saw today. In the booth, we can also tell you more about our fast discovery framework that we use to identify and prioritize AI opportunities. And finally, there is a Crack the Code competition. So hop over to our booth to start a conversation with myself or my colleague Alice, who will be there, and we can provide you with the code. So thank you, and back over to you, Joel.
Thank you so much, Kristien, and to your team at Radix. What a compelling overview. And as I said, I learned so much from what you had spoken about, what Wim spoke about earlier. I will say, many of those similar methods, when you're looking at the Petri dish, we call that remote sensing and, of course, image recognition. We are using those exact same algorithms and those exact same methods, albeit at a very different scale, from satellite mapping for the United Nations, to take those very menial tasks. It used to have to be humans. I remember working on Ebola 2014, and we were mapping the roads, mapping the clinics. Now the AI can be a real boon to help get through the boring stuff. And what you're doing at the nexus of pharma, especially during this global pandemic, is so timely. And again, thank you so much. And we really learned a lot from your presentation. For colleagues